Welcome to this explainer. Today, we are going on a truly fascinating journey together. We're going to explore how a natural, microscopic, glowing bacteria has been entirely transformed by scientists into a high-tech tool for crime fighting and environmental protection. I know, it sounds a bit like science fiction, but it's incredibly real. It's happening right now. And it's all thanks to something called optical biosensors.
Okay, let's dive right into this. Here is our roadmap for today. We'll start with the magic of bioluminescence and the chemistry behind the glow. Then, we'll look at coupling reactions for detecting ethanol, how we catch explosives with light, testing water pollution with microtox, and finally, measuring toxicity sensitivity. Let's get started.
Section 1. The magic of bioluminescence.
So, before we can even think about building a high-tech sensor, we really need to understand the mesmerizing natural phenomenon that makes all of this possible. Take a look at this nutrient-rich broth glowing with that brilliant blue light. That light is sitting right around the 500 nanometer wavelength. And the crazy part? It's being produced entirely by a living organism called Bacterial Luciferase. What's so amazing about these luminous marine bacteria is that they don't just glow randomly. No, they produce this light only upon reaching a critical population size. It's a built-in natural biological trigger.
Moving on to Section 2, the chemistry behind the glow.
But how exactly are they pulling this off? Let's peel back the curtain on the invisible molecular machinery powering that visible magic we just saw. Unlike a lot of other glowing creatures out in nature that rely on chemicals called luciferins, Bacterial Luciferase operates a little differently. It uses a very specific bacterial reductase process. In step 1, a reduced molecule called FMNH2 forms an excited complex. Step 2, this FMNH2 reacts with oxygen and an enzyme called Bacterial Luciferase. And finally, step 3, this reaction triggers a sudden release of energy, which we actually see as visible light, emitted between 478 and 505 nanometers. So, the absolute key here is that this specific combination, FMNH2, oxygen, and luciferase, is the unique chemical engine creating our biological light bulb.
Section 3, coupling reactions, and how we use them for detecting ethanol.
Now you might be wondering, how do we actually use this natural biological light bulb to detect invisible chemicals in our everyday world? Well, what's really interesting about this slide is how it illustrates a coupling reaction. This is where an invisible chemical, like ethanol, gets connected to visible light. Here's how it works. If we want to measure ethanol, we oxidize it. Converting ethanol to aldehyde reduces a molecule called NAD+ into NADH. That NADH is then used to reduce FMN into FMNH2. And if you remember from our last step, FMNH2 is the exact fuel our bacterial luciferase needs to emit light. It's honestly incredibly clever. We've created a chemical cascade where more ethanol directly fuels the creation of more FMNH2, which basically means a brighter light. We just measure the light photometrically, and boom, we suddenly know exactly how much ethanol is present.
Section 4. Catching explosives with light.
We are escalating things pretty quickly now, going from measuring alcohol to the high-stakes world of forensics and national security. It's quite a stark contrast, isn't it? On one hand, you have these microscopic, peaceful marine bacteria just glowing away in the ocean. On the other hand, we have massive macro-level threats like TNT, a highly volatile explosive. But believe it or not, the exact same bioluminescent principles we just learned can actually be used to detect the presence of TNT via a highly sophisticated bioluminescent immunoassay.
To understand how we actually catch the TNT, we first need to wrap our heads around the concept of a turnover number, or TON. This is defined as the number of moles of substrate that a mole of catalyst can convert before it becomes inactivated. Basically, think of an enzyme like a tiny biological factory. The turnover number tells us exactly how many products that little factory can pump out before it has to shut down. The higher the number, the more powerful the biological amplifier. And that brings us to this absolutely massive number, 70,000. In our TNT detection system, scientists link the explosive to a specific enzyme called glucose 6-phosphate dehydrogenase, or G6PD. G6PD has an insanely massive turnover number. Just one single molecule of this enzyme can produce 70,000 molecules of product per minute, so it acts as a phenomenal, high-powered amplifier for our entire biosensor. Looking at this immunoassay process, you can really see the brilliance of the design.
By binding the TNT to our G6-PDD enzyme factory, the presence of even a microscopic amount of TNT triggers this massive amplification process. The enzyme rapidly pumps out the fuel needed for the bioluminescent cascade, which results in a sudden, highly measurable output of light. We are literally using biological light as a giant red flag for the presence of trace explosives. It's wild!
Section 5. Microtox Testing Water Pollution.
Let's shift our focus away from forensics now and talk about protecting our planet. Because here, our glowing bacteria step into the role of environmental guardians. This is the Microtox Testing Unit. It's an in vitro testing system that utilizes our bioluminescent bacteria to detect toxins in the air, water, soil, and sediment. It's what's known as a metabolic inhibition test. So instead of looking for one specific chemical, like we did with ethanol or TNT, Microtox actually uses the bacteria to tell us about the overall health and safety of an entire environmental sample.
It's a really stark visual. Over on the left, in the clean water, we have healthy, bright, beautifully luminescent bacteria. But when these bacteria are exposed to toxic materials, their normal metabolic processes are inhibited. A sick bacterium just can't run its internal machinery properly, meaning it stops producing that crucial FNMH2 fuel. The result? The light fades. By simply comparing the bright tubes of healthy bacteria to the dimming or completely dark tubes of sick bacteria, scientists instantly know if a water sample is polluted.
Now, there are a few key reasons why this particular biosensor system has become the universal standard across the globe. First off, it's extremely rapid, giving accurate results after just a 5 to 15-minute exposure. It's also super cost-effective since it only needs tiny sample volumes. It has incredibly broad applicability. You can use it for water, soil, sediment, and crucially, it's highly reproducible worldwide. That means a scientist testing water in Tokyo and a scientist in Toronto can look at their data and compare it with absolute confidence.
Section 6. Measuring Toxicity Sensitivity.
Okay, so we know the light dims when there is pollution. But how do environmental scientists mathematically quantify the exact danger level of a sample? Well, they use a standard metric known as EC50. This stands for the concentration of a toxicant that produces exactly a 50% reduction in the light emitted by the bacteria. It's the ultimate benchmark. If it takes very little of a sample to cut the light in half, that sample is highly toxic. If it takes a huge amount of the sample to dim the light, the toxicity is relatively low.
So, knowing what EC50 actually is, how do we visualize it? How do we map out this mathematical relationship between the poison in the water and the fading light of our bacteria?
And this brilliantly illustrates the answer right here, a classic dose-response graph. On the bottom axis, you have the concentration of the toxicant, that's your dose. On the vertical axis, you have the percentage of bioluminescence inhibition, that's the response. As the concentration of the toxicant goes up, the inhibition of the light sharply increases right along with it. The math is just undeniable. By plotting this curve, researchers can perfectly pinpoint that EC50 mark, providing governments and environmental agencies with exact, hard data on pollution levels.
Well, we've reached the end of our explainer, but I really want to leave you with this final thought. Nature's microscopic warning system. Isn't it profoundly amazing that humanity managed to take a tiny, naturally glowing marine organism, completely map out its biochemistry, and harness it to create the ultimate biological detective? I mean, from catching microscopic traces of explosives to acting as the gold standard for protecting the very water we drink, optical biosensors prove that sometimes the absolute most powerful technological advancements are just waiting for us out there in the natural world. It really makes you wonder, what other biological miracles are out there, just waiting to be transformed into tomorrow's life-saving technology? Thanks so much for joining me on this deep dive.